Welcome to MetPhys 2020, day 19. We've talked about this every day. It's cost effective, it's green, and it's inclusive. It's not just inclusive for you listening at home, it's also inclusive for the speakers. We've pre-recorded a talk from a speaker who otherwise wouldn't have been able to participate due to her own personal responsibilities at home and in this crisis. And I think that's fantastic. We can make this inclusive not only for people who can listen to it anywhere, anytime, any place, but also the speakers who might not have been able to come to a meeting or who might not have been able to speak can record their presentations at a different time that's convenient for them so that you can all hear about the great work they're doing, that they can be involved. And I think that's a beautiful thing about this new model for science conferences. Cassidy has nailed it again. She is really killing this Inception challenge. I'm looking forward to the last day of the meeting where I'm going to put every single one of these 24 photos that she has taken side by side. I think it's going to be a really nice piece of art almost that we can enjoy. So thank you, Cassidy. Right now, take your photo. Thank you. Yesterday, I launched a little bit of a secret that there will be a secret speaker participating on Tuesday of next week during Ebru Abai's presentation. If you can take a few of the clues we're putting out clues every day to guess who that will be. I'm going to reveal who it's going to be on Monday, so you all have a chance to share that and make sure everyone's tuning in. It's going to be really exciting. The clue number one that I shared yesterday was serendipity. These are not meant to be easy, and if you look closely at the continuing presentation, you may get another clue about what that refers to, which might lead some of you to know who the speaker will be. If you have an idea, DM me, don't ruin it for everybody. But if you have no clue who it is, feel free to tweet it into the wild. All right, so how do we do question and answer time? We do this on Twitter. We use the MetPhys 2020 hashtag to link our questions, and we also at the speaker. Today's speaker is Andre Marette. His handle is very simple. It is his name. So if you put the speaker's name or the hashtag MetPhys 2020, we will all be able to follow along and see what's going on. If you put both, that is best. Do this during the talk. After the talk, you can ask as many questions as you want. Yesterday, we had a fantastic back and forth for a few hours with Ali. Her talk was amazing, and there was a lot of excitement. We've had that for a lot of the speakers, and I know they enjoy it. If they don't reply immediately, they may be feeding their children or attending to other duties. But the nice thing about this is they will be able to answer those questions and get back to you. And what's really nice is we're all able to read those answers and potentially use them as a resource. I have noted a few people who've watched these videos on YouTube a week after the talk are starting to ask questions, and I think that's really fun. So enjoy that. If you are not on Twitter, I think you can put it into your Safari or Explorer page and go to the search bar and just type MetPhys 2020 without even having an account. If you do need an account, you don't have to make it personal. And you don't even need to like or follow anyone. You can just search for the hashtag. So enjoy. It's a really nice feature of this space. And I think what I'm really enjoying is what a lot of people are sharing as part of the community. If you share something unique and you tag it with MetPhys 2020, you're liable to win a MetPhys 2020 prize. Of course, those prizes don't exist, but you know, you can still claim them and take the pride that goes along with that. I've said this every day. We need to be grateful to the speakers. Without the speakers, this wouldn't happen. And I can tell you that all these people, especially on this front page, replied to me very rapidly when I launched the idea to launch this program. Within 1 week, we had a full program up until today. And then we've been able to expand it to April 24, at which point MetPhys 2020 will come to an end. Don't worry, there will be MetPhys 2021, and we already have a lot of great speakers lined up for you for that. If you do know of a young colleague who is in need of a little bit of exposure, we have a couple of slots left. I do encourage you to share with those persons who may be heading to the job market or have just started assistant professor positions. I think it's really nice that we've been able to feature young scientists in this program, but of course, young scientists doing some of the best work around. Today's speaker is Andre Marette, who was my graduate mentor. He's also a close friend. There are other speakers for the next 2 weeks, and here is a clue about that secret speaker. Can you guess the secret scientist? And go ahead and see if you can figure that out. If you want to thank me personally, the Pathway to Stop Diabetes program from the American Diabetes Association really launched my career. I know it's also been a support for Celine Riera, who's going to be speaking later in the program. If you have some spare funds available and would like to make a donation to a good cause, this is a great one, and it would really honor us if you did indeed go ahead and do that. If not, that's no problem. Enjoy the talks, enjoy the resources. Don't forget, you can share these links. You can share the YouTube link and the Facebook links with your friends and colleagues, those that might not be on these platforms, so that they can enjoy them, share them with your students. These videos are going to be up there for a long time, and we're glad that we can provide this resource. So today is Friday. I only found that out yesterday. So our next talk will not be tomorrow, the next day, but the day after. If you count the sunrise twice, then we're ready to go. We're going to hear a fantastic talk from Alex Muir in Chicago, where he's going to talk about what's to eat around here, nutrient availability in the tumor microenvironment. So make sure to tune back in Monday for that. Watch out over the weekend using the Twitter MetPhys2020 hashtag. I will be launching more clues about our secret speaker for next Tuesday. And Monday, I'll reveal who that is. So enjoy. I wonder if anyone's going to be able to guess it. I think the clues are going to get easier day by day, but that may not be the case. I might keep them cryptic. All right, so without any further ado, I'm going to transfer host over to Andre. And enjoy the talk. I'll see you in the Twitter space. Make host. Andre, I'm making you the host right now. Stopping my share. Stopping my video. All right, good. We can see you, Andre.
Okay. I'm going to share my screen. Where is it? This is the one here. Okay, I hope everyone can see my screen. Okay, so Phil just confirmed that you can see and hear me, so that's great. Just going to put that on the slideshow now. And first, I'd like, of course, to thank Phil for putting this together. It is an amazing initiative. I've been listening to many talks. The quality is amazing, and a lot of people I've challenged myself because they showed a lot of non-published data. And so today, what I'm going to show you is all unpublished data except the last 10 minutes. But these data have been published last month. So most of it has been either published very recently or being unpublished at all. So yes, I'm going to talk about bacterial translocation to tissues, metabolic tissues in humans. But before I'm going to tell you about that story, I have another tale to tell you about. It is the effect of dietary proteins on metabolic syndrome and the implication of the gut microbiome into this effect. So for those of you who knows me, you know I've been working a lot on the origin of inflammation. We all know that inflammation is a common denominator into favoring or producing metabolic disease and cardiometabolic disease in general. And of course, we all know that the expansion of the adipose tissue in obesity promotes inflammation, and that this inflammation will cause, of course, insulin resistance in liver and muscle, favor also the development of liver disease, will reach the beta cell and favor the development of high left inflammation and type 2 diabetes. And the spreading of inflammation, of course, as we all know, will also promote vascular dysfunction, atherosclerosis. So inflammation is involved in insulin resistance, type 2 diabetes, but also in cardiovascular disease and heart disease. So I spent a long time, including some studies I did a long time ago with Phil when he was a student in my lab, to try to understand the regulation of adipose tissue inflammation. But in recent years, it became obvious that another major organ was involved into the origin of inflammation and obesity. And it has been overlooked for so many years, the intestine and the gut. And of course, the reason why it's now looked more in terms of playing an important role into driving inflammation and metabolic disease is because of these bacteria, the gut microbiome, the gut microbiota, of course, which is now known to be an important player. Some people even call it an organ that is involved into promoting insulin resistance and type 2 diabetes. The reason why the gut microbiota, the intestine, is so important is not only because of the gut microbiota. Of course, studies for many years have shown that the gut produces many metabolites, hormones, factors that are regulating metabolism. But what is driving inflammation from the gut has been shown to be many years ago by Patrice Cani and Eric and Burcelin and others, that it is the lipopolysaccharides that are released when there is a leakiness in the gut, so this reduction of gut integrity, and therefore LPS, this bacterial fragment from gram-negative bacteria, and can cross the intestinal barrier and promote metabolic inflammation. So this is the new term that was invented many years ago by Kenny and others, metabolic inflammation, which is of course the same as low-grade inflammation and obesity. So with this background in mind, we've been, of course, quite interested about the microbiome, this mutualistic relationship we have with our microbes. I think most of you are very familiar with the fact that we are not alone, is that if you compare to the human genome, the metagenome, which is the genome of the bacteria and other microorganisms that lives with us, is quietly, much more important in terms at least of expression of genes and even in the number of bacterial cells or other type of microbial cells. So we are indeed in symbiotic mutualistic relationship with our microbes. And it has been quite amazing to see the recent development in this field, where we are understanding better and better the role of individual bacteria into driving metabolic processes, and unfortunately with pathogenic bacteria driving metabolic diseases. So what really brought me into the field, because as many of you, I had some skepticism about the role of the gut microbiome into driving metabolic disease. There was many studies, probably around 2010, 11, that were started to show that you can transfer metabolic phenotypes from the gut microbiota from a donor mice to a germ-free mice. And this study is probably the most important one, because they were able to show that even if you take identical twins, so genetically identical twins, obviously they were not coming from the same family, they were separated when they were very young. So you have the lean twin, when you transfer the gut microbiota from the lean twin to a germ-free mice, of course the mice will remain relatively healthy and not fat. But if you transfer the gut microbiota from the obese twin, you will get increase in fat accretion in this mice, you will get obesity and a reduction or impairment of metabolism. So you can transfer metabolic phenotypes from the gut microbiota, and you see differences even when it's coming from identical twins. It doesn't mean that genetics are not modulating how the gut microbiota will impact metabolism, but certainly you can drive this phenomenon even with getting the microbiota from two identical twins, so genetically identical persons. So of course, as I said before, we want to understand how the gut microbiota is promoting metabolic disease. As I've mentioned already in the previous slide, we think that there's a gut inflammation that is driving meta-inflammation in the metabolic organs, such as fat and liver, and probably also muscle and the islets. And this is very important, of course, and this is again the work of Patrice Cani and his collaborators in the between 2007 and 2009. What we know now from the work of many labs, because now this is really a trendy field, is that several factors are controlling the gut microbiota. Of course, those dietary factors are probably the most important regulator of the gut microbiome, but host immunity and infection, host genetics, as I mentioned a few minutes ago, but also in particular drugs and xenobiotics are all able to influence the gut microbiota. All human studies so far have shown that after dietary factors, probably a drug that have been used by participants in studies have been one of the most powerful regulator of the gut microbiome. But of course, diet is not the only environmental factor that will control the gut microbiota. There are other exposome factors, diurnal patterns, climate, light, temperature, microbial exposure, of course. All these things will influence the gut microbiome over time. And this is, I added a lot of arrows here, not to highlight the important role of the gut microbiota everywhere in metabolic homeostasis, but just to make sure I was going to beat Phil's White for having used the most numbers of arrows in a single slide. So sorry, Phil, I think I'm going to win that challenge. Of course, I'm quite interested into understanding the role of the microbiome in driving the effect of different dietary components, of course, macronutrients. But I just wanted to point out that our team has been playing an important role with others into looking at the impact or the role of the gut microbiota in driving responses to polyphenols. Of course, we all know that fibers are important prebiotics, but it has been recognized recently that polyphenols in fruits and many other types of food can also make big changes in the gut microbiota. And you can actually transfer this phenotype by a transfer of the gut microbiota. So fecal transfer, fecal microbiota transfer can actually reproduce the phenotype that you can get by feeding dietary polyphenols. And the way these polyphenols and the gut microbiota are interacting, there are several ways. Of course, the gut microbiota will metabolize the different type of polyphenols. For example, in berries, you have proanthocyanidins, L-argitannins, and the very famous resveratrol. All these molecules can be metabolized by the gut microbiota, and of course, there will be secondary transformation in the liver. So this is already one interaction between the gut microbes and polyphenols, but we have shown in the past that many types of polyphenols can actually drive the increase in Akkermansia muciniphila, which is a very important bacteria that has been involved into healthy phenotypes in metabolic homeostasis. So this is a prebiotic effect of dietary polyphenols, this raising of Akkermansia muciniphila. We'll talk about this bacterium a little bit more, well, actually quite a lot after that. There's, of course, a community prebiotic effect because polyphenols in general will increase the richness or the diversity of gut microbes, and this is in general associated with health. And finally, there's been quite a number of studies that have shown that polyphenols can increase also gut barrier, gut integrity through driving pathogenic defense mechanism. So all of these different mechanisms will, of course, lead to improvement of insulin sensitivity, a reduction of glucose intolerance, a reduction of inflammation, especially metabolic endotoxemia, and reduce hepatic steatosis and inflammation. And overall, this will reduce the metabolic syndrome, liver disease, and cardiovascular disease risk. And as I said before, most of these phenotypes, these beneficial phenotypes of polyphenols given in food can be mimicked by transferring the gut microbiota from a donor mice to a naive germ-free mice. So colonizing with these bacteria will reproduce the phenotype. Talking about Akkermansia muciniphila, the discoverer of this bacteria is Patrice Kenny, and Patrice Kenny recently published a paper last year in July in Nature Medicine showing for the first time that you can supplement obese human volunteers with Akkermansia muciniphila, and you can actually increase their metabolic homeostasis, meaning that they have seen an improvement of insulin sensitivity in these individuals. So not only Akkermansia is safe in that particular proof of concept study, but it was able to improve insulin sensitivity and reduce systemic inflammation. What is more striking about Akkermansia as well is that you can neutralize it by pasteurization, and you can still maintain its biological activity, which led to the concept that I've written with Fernando Ani and Jonathan Trezor from Hamilton University, McMaster University, this concept that postbiotics could be used to treat metabolic disease. So what Patrice Kenny has shown is that, of course, live Akkermansia muciniphila given as a probiotic can improve metabolic health, have metabolic benefits in these obese individuals. That has been found in mice, of course, but also in humans. But after neutralization, components of muciniphila and even the bacteria coming from the membrane of the bacteria can actually reproduce the effect, and sometimes even better than Akkermansia per se. So this is leading to the concept that there might be bacterial targets or bacterial components, proteins or others that can actually be used to treat metabolic disease. And this is a very new concept that has attracted a lot of attention recently, including the attention of several members of my own lab. Now, to put you in the context of the first tale I want to talk to you about today, the first story is that, of course, we all strive to work in humans and try to understand why obesity is associated with metabolic disease and what are the original mechanisms that are causing this defect. And most of us are using either cellular or animal models. And of course, we're trying to mimic the human conditions. These are models, experimental models. But if you work with models to study human disease, you have to try to make it as best as possible as a model. So common models that have been used by many of us is, of course, genetically modified animals. Think about the OB or DBDB mice, for example, which are models of obesity and diabetes in rodents. But of course, many of us are using diet-induced obesity. So we are giving high-fat, high-sucrose diet to promote obesity within a few weeks or months into animal models, rats and mice. So for many, many years, I've been working on such models, including with Phil White and many other students and postdocs in my lab. And something has been really in the back of my mind for so many years, a frustration, so to speak, is that while most of us, or most studies are focusing on fat and carbohydrate into driving metabolic disease and try to reproduce again a human disease, because I don't see many wild mice developing obesity, right? It's mostly humans. So why don't we focus more on dietary proteins? There could be several reasons for that. It's not easy to work with proteins and so many amino acids, but to me that does not justify the lack of effort to understanding the role of proteins. And if you're like me, most of us, and including myself until recently, when we were using diets to promote insulin resistance and obesity, we were using different carbohydrate sources, different, of course, lipid sources as well. We would use lard and corn oil, etc. But in most protocols, people would use only casein, a single protein. This is, of course, crazy if you think about it, because humans are not eating only casein. We're not even eating only milk or milk products, right? We're eating probably 20 or 30 different types of protein coming from different type of animals. Animal proteins, et cetera, are represented here. So we've been, thinking for a long time, what is the role of dietary proteins in driving metabolic disease? And if we have different effects of different proteins, is it coming from change in the gut microbiome? My lab has shown more than 20 years ago already that if you only switch casein for fish protein or soy protein, you can have a major impact on insulin sensitivity. You can have a major impact on the development even of adiposity. But in these cases, we were changing one protein, casein, for another one. What we wanted to do in the studies I'm going to show you today is why don't we try to compare casein versus a dietary, versus a diet that will contain a large variety of protein sources. So this is what we did in that study. So the objective of that study was to evaluate the impact of a dietary protein mix that is more relevant to typical intakes of proteins in humans on weight gain, metabolic health, and gut microbiota in an obesity mouse model. And these, as you will see, are not very easy study to do. Just to make the diets, it can be a nightmare. And so I needed to convince two very bright students to take this challenge And Beatrice and Noemi, yeah, you can see their picture here, really took it and did it, and they nailed it, as you will see in a few minutes. So thanks for their incredible work in the last 3 years to actually get this project done. So the first thing that we had to do, and that was the work of Noemi especially, was to design new diets. And it looks quite easy, but it's not easy at all. So most diets we use to promote obesity contains, of course, a lot of carbohydrate and fat and some proteins, as shown here. So 50% fat, 35% carbohydrate, as compared to 75% carbohydrate and 10% fat. What is usually stable in all these different diets is the amount of proteins, 15%, which is what our vet wants in our animal facilities. And this is recommended by all the suppliers of food, animal food. So the design of the study was, so this is the typical whole casein dietary protein. So in that particular diet, all of the proteins were going to come from only casein, versus using these different type of animal sources to provide a much more mixed source of proteins. And we didn't take this percentage by coincidence. This is coming from USDA human data. So we look at what people in the US are eating in terms of protein, and we try to reproduce that as best as possible in that diet. So this is a kind of a humanized diet, at least for the protein component of it. And so it contains animal protein coming from beef, pork, chicken, of course, some dairy, casein and whey protein, and fish, cod, and also soy, pea, rice, and eggs. So this is the proportion of what people are eating in the US. So not obese people in general, the population in the US. So you see very, very different diet in terms of the composition of the protein. But the challenge, of course, is that when you get beef and pork and chicken, you don't get pure source of protein. We were able to find companies that were selling us extracts that were containing mostly proteins, but most of them also contain some level of fat. So we had to analyze, Noemi had to analyze the content of fat in all of these different dietary sources, and to be able to build these diets to make sure they were equivalent in terms of macronutrients. So not only in terms of protein, but also in terms of fat and in terms of carbohydrate. And she was able to do that. She was able to balance the diet. So we have the low-fat diet, low-fat, low sucrose. We didn't use a chow diet in that particular study, of course. We used low-fat, low sucrose, and containing either casein or the protein mix. And we have the high-fat, high sucrose, casein, and the high-fat, high sucrose containing the protein mix. So, of course, the high-fat contains more fat and more carbohydrate in terms of, not more carbohydrate, but more sucrose as compared to the low-fat. But you see the difference between the casein and the protein diet. There's no difference in terms of protein, in terms of carbohydrate, in terms of fat. We were very good. Noemi was very good to balance even the amount of saturated, monounsaturated, and polyunsaturated fat between these two diets, or these two diets here. And the amount of fibers also is constant. So the only real difference between those diets is the composition of the amino acid into the protein. And I'm going to show you that a little bit later. So this is the protocol. We use male C57 black cis mice. There were 4 groups again, the 2 low-fat animals, so not becoming, of course, the healthy controls on the casein or the protein mixed diet, and the high-fat, high sucrose-fed animals on the same type of dietary proteins. So the mice arrived, then we kept them for 2 weeks on the low-fat, low sucrose casein diet. At day 0 of the protocol, we started them on their dietary treatments. We took some fecal samples before and during treatment, and we take blood sampling at different time as well. We did an OGTT at week 11. The protocol was lasted for 12 weeks. And at the end, we did a fasting to take blood samples. And of course, we did some insulin injection to measure insulin signaling in metabolic tissues from these animals. And then we euthanized the animals, and we took all the tissues we could to study the metabolic phenotypes of these animals, as well as to do insulin signaling determinations. So that's the first set of the results. So rapidly, what you can see is that the animals on the high-fat diet, so the blue squares were, of course, taking more weight during the 12 weeks as compared to the animals on the low-fat, low-sucrose diet. This was expected. Those on the low-fat, low-sucrose diet, irrespective of the dietary protein sources, we could see no difference in body weight gain. And this is the final body weight gain at the end, so no difference. However, the animals that were put on the protein mix diet, more representing the human dietary intake, protein dietary intake, you can see that these animals put up more weight starting approximately at week 6. And as you can see, the final body weight gain was higher, greater in these animals. So just changing the casein to the mix of protein, again, it is isoprotein, isoenergetic, and everything. The animals put on more weight because of the change in the dietary composition in terms of protein only. These animals are eating the same amount of fat and sugar. It is only the protein component that is different. And again, the amount is the same, it's the composition that is different. These changes were not caused by difference in food intake. Of course, the animals on the high fat, high sucrose per gram had less food intake because these, of course, these diets are more rich in energy. But the difference we saw in the body weight gain were not explained by change in food intake. The fat mass was different, of course, over time. Very small change in lean mass. As you can see, a small increase in gastrocnemius muscle, which is interesting because, of course, when you give a more diversified source of protein, you can think it's probably better to build muscle mass. So we did find that. But most of the changes that we saw in the weight were due to the increment or the increase in visceral fat in the PM diet as compared to the casein diet, again, in the high fat, high sucrose background. Inguinal fat was not different between the two high fat fed animals. So this is mostly a change in the level of visceral fat, which was expected. Interestingly, we also find a small increase in brown adipose tissue. And I've worked many years on brown fat. I know when brown fat is getting a little bit bigger, it's probably that the tissues is keeping more fat storage. So there's less activation of brown fat. And we didn't look into that in a lot of details, but we still look at UCP1 gene expression, which is the best marker of activation of brown fat activity. And as you can see in the PM diet, the animals had less UCP1 as compared to the casein diet. And this is also consistent with the fact that SID1, the iodinase 2, the rate limiting enzyme for thyroid hormone production, and PGC1 alpha were all reduced in this particular group. And this is, of course, very important for the programming of UCP1 and brown fat activation in that tissue. So we believe that some of the weight gain in these animals is probably due to reduction of brown fat activity, but that has not been demonstrated by measuring, for example, brown adipocyte respiration or thermogenesis per se. We didn't put animals in metabolic cages yet. So what about glucose homeostasis? So this slide is showing you at the end of the protocol, the fasting glycemia. It was increased, of course, with the high fat, high sucrose fed animals, but no significant difference between the two sources of protein. However, when you look at the oral glucose tolerance, you can see that the animals on the mixed protein diet were clearly more glucose intolerant. And this is also seen at the area under the curve of these glucose excursions. No effect on the low fat, low sucrose diet, however. So you need a combination of a high fat, high sucrose diet to see this added impairment of glucose tolerance that is driven by the mixed protein as compared to casein. Is that associated with a change or impairment as well of insulin sensitivity? Of course, when we look at fasting insulinemia, a hallmark, of course, of insulin resistance, you can see that the PM-fed animals on the high-fat, high-sucrose background diet, again, had more insulin in the fasting state, and they release much more insulin to cope with the glucose challenge as compared to the animals that were fed the casein diet. Again, no change in the animals that were lean and kept on the low-fat, low-sugar diet. But on the high-fat, high-sugar diet, you can clearly see that there's a big increase in insulin secretion, which is in very good relationship or agreement with the impaired glucose tolerance. This is at least partly related to the increased insulin secretion. We were able to measure C-peptide levels at 0 and a 15 minutes time point, and you see the elevation is well matched with the increased insulin levels that we found during the glucose tolerance curve. So it's probably at least involving changes in insulin secretion, but we haven't measured hepatic insulin clearance, so that might also play a role into these effects. So we had access to tissues from these animals. Some of them were injected with saline or insulin to measure the insulin-induced activation of this pathway. And the reason we did that, of course, is that many years ago, it's like almost, well, 20 years, 10 to 20 years ago, we have made some interesting findings about the role of proteins and amino acids in driving a very important pathway, the mTOR S6 kinase pathway. What we have shown in those papers is that in the presence of hyperinsulinemia and obesity, an increase in some proteins, especially amino acids, was driving the mTOR pathway S6 kinase 1. These are serine threonine kinases. And what we have shown in many of these papers, in adipocytes, in muscle, liver, but also in tissues from human individuals that were insulin resistant, were shown that overactivation of this pathway was driving RS1 phosphorylation on serine residues, and in particular serine 1101, which is a known target of S6 kinase 1. So this is a feedback mechanism that is driving insulin resistance in obesity in both animal models and in humans. So we wanted to know whether this pathway, which is responsible for, of course, inhibiting PI3 kinase and AKT activation of glucose uptake or glucose metabolism in tissues, was involved in the story with the mixed protein. So Vanessa Hood, a very talented professional assistant in my lab, did some signaling work. And as you can see, looking at the phosphorylation of S6 uses a readout of the mTOR S6 kinase pathway was really increased, as you can see here. And I think I have the quantification here. So you can see that the phosphorylation of S6, so the activation of the pathway was increased, of course, by obesity, but it was increased even more in the context of feeding the protein mixed diet as compared to casein. And you have, of course, the effect of insulin. So you have minus plus insulin in all of these different graphs. The insulin is driving this. In the presence of the mixed protein, you have also this activation, which is greater than in the low-fat, low-sucrose PM, but also as compared to the high-fat, high-sucrose casein. And the phosphorylation of RS1 on serine 1101, this marker of S6 kinase 1 activation, was also markedly elevated, again, in the context of feeding the protein mix as compared to casein. We have a significant difference. And this was associated as well into reduction of AKT phosphorylation as shown here. So AKT phosphorylation also is markedly depleted in the animals fed the dietary protein mix as compared to the casein. And we also measured PKC delta. PKC delta is another kinase that have been shown to phosphorylate RS1 on this particular serine residues. And this protein was also more elevated in the liver of the animals fed the high-fat, high-sucrose protein mix. So I should have said from the beginning that those are liver data. We focus on the liver first, and which is confirming, therefore, that in the liver, there is increased activation or overactivation of mTOR S6 kinase pathway, which is driving hepatic insulin resistance by inhibiting PI3 kinase pathway as measured by the readout of PI3 kinase, here the phosphorylation of AKT. So this was found in the muscle and in the liver, I'm sorry. We've done exactly the same studies in liver. And I will go very quickly because basically we didn't see that at all. So we measured RS1, PKC theta, the phosphorylation of RS1 in serine 1101, the activation of the mTOR pathway, and phosphorylation of AKT as well. We were not able to detect any significant difference between the animals fed the high-fat, high-sucrose diet on either on the casein or the protein mix. The only thing that remained significant in muscle was the overactivation, or the overexpression, I should say, of PKC theta, but this did not result in a significant elevation of phosphorylation of virus-1 on the targeted serine 1101. But however, because there's a tendency, one might think that if we would wait longer, maybe the same phenomenon would have been seen in muscle. But after 12 weeks, the only tissue where we could detect this difference was in the liver. We haven't looked at the adipose tissue in much details, however, but at least the muscle doesn't seem to be affected as much as the liver. Of course, the primary objective or hypothesis behind this work was that the dietary proteins might actually regulate metabolic homeostasis to change in the gut microbiota. So we use 16S RNA sequencing of the fecal materials from these animals. So you have all the groups here, you have all these small squares, all represents a single animal. So you have the low-fat casein, the low-fat, low-sucrose protein mix, and you have also the group of animals on high-fat, high sucrose with the casein or the protein mix. And of course, we have measured a bunch of bacteria. These are only those that are represented here are those that were changed the most or were most abundant in these fecal samples. But just to show you an example, for example, Intestimonas here was clearly overrepresented in the animals fed the protein mix as compared to the animals on the casein mix, on the casein protein. And of course, the animals were both on high-fat, high-sucrose diet. So some of these changes that we see in the microbiome are not related at all to the high-fat, high-sucrose diet. Of course, if you start comparing low-fat, low-sucrose with high-fat, high-sucrose, you will see a lot of microbial, a lot of gut microbiota changing, of course, because of the effect of high-fat and high-sucrose. But for the purpose of my talk, I'm going to focus on those that have been only regulated by the dietary proteins in the context of a high-fat, high-sucrose diet. And those genera that we found to be particularly interesting because they were significantly affected by the protein diet have been labeled here with these arrows. So, for example, Adelclorsia, as well as Tizarella and Intestimosas were increased by the dietary protein mix, whereas Bacteroidetes, as well as Akkermansia, were decreased by the protein mix. And of course, what caught our eye from the beginning was Akkermansia, because of course, we are now investigating the role of each of these bacteria in producing the metabolic phenotype we have seen. But for Akkermansia, we already knew that this was involved in metabolic homeostasis. So we just wanted to confirm rapidly Akkermansia muciniphila. And indeed, we can see that in animals fed the protein mix diet, when we use PCR, we see clearly a major reduction, a significant reduction of Akkermansia muciniphila abundance in the fecal microbiome. But this was also found in the low-fat, low-sucrose background. So a single reduction of Akkermansia muciniphila probably cannot explain the metabolic phenotype of having more glucose intolerance in the high-fat, high-sucrose background, because in these animals, of course, there were no metabolic defect. So it's just to say that Akkermansia muciniphila reduction is probably one of the mechanisms that is driving such a response in the context of a high-fat, high-sucrose diet. And just to give you an example where sometimes looking at the diversity of the gut microbiota is not associated with a better metabolic health. Of course, when you start feeding animals with a mixed protein diet, you will get a much more diversified microbiota, as shown here by the Shannon index. The Shannon index is a very good marker of the alpha diversity. And you can see when we feed animals with the protein mix, we have a more diversified microbiome because, of course, the microbes that are there are driven by different type of proteins. And in that case, of course, what we have seen is a reduction of metabolic health. So there's a lot of cases in the literature now showing that alpha diversity or the diversity of the gut microbiota is not necessarily associated with metabolic health. It depends on the particular conditions, it depends on the treatment, it depends on many things. It's interesting, of course, to look at the composition of the gut microbiome, but it's also interesting and very important to look at the function. So to start to look at the function of the gut microbiota, we begin to measure the potential impact of the different protein sources on microbial products. And one of the most well-known products of the gut microbiota, of course, is the short-chain fatty acids. And most of you familiar with gut microbiome work, you know that these three short-chain fatty acids, acetate, butyric acid, and propionate, are the major short-chain fatty acids, and are coming from the fermentation of fibers by the bacteria. And as shown by many labs before, including our labs, in obesity, if you compare, for example, the high-fat, high sucrose casein with the low-fat, low sucrose casein, usually the short-chain fatty acids are reduced. And that's one of the hallmark of obesity. However, what you can see, what you can appreciate, is that those short-chain fatty acids are increased by the mixed protein. So this is not an increase by fibers. The amount of fibers was exactly the same in all those diets. So by itself, the protein mix is increasing short-chain fatty acids. But remember, these animals, especially in the context of high-fat, high sucrose feeding, have a decreased metabolic health. They have an impairment in glucose metabolism and insulin sensitivity, yet they have more short-chain fatty acids. What is more interesting to us is that the so-called minor short-chain fatty acid, minor because they're less abundant than the major ones, also called the branched-chain fatty acids because they're coming from the proteolytic fermentation of branched-chain amino acids. These we really wanted to look at them because, of course, we're looking at the impact of different types of proteins containing branched-chain amino acids, of course. And this is quite interesting because, especially in the context of obesity, in the animals that were given the high-fat, high sucrose diet, I think you can really appreciate that for all of them, isovaleric acid, isobutyric acid, and valeric acid, there is an increased production in the fecal samples of the animals fed the protein mixed diet. This is highly significant, especially for isovaleric acid and isobutyric acid. If you look in the literature, there's very little known about the actual production of branched-chain fatty acids and their role in metabolism. Very little work, which was to us very intriguing and very interesting to pursue. So this was, of course, the impact of what we call the long-term protocol. 12 weeks of feeding these diets. So we have change in the gut microbiome, which is associated with change in metabolic health in these animals. So there is an association there, but of course, it's not a cause and effect demonstration. If you want to demonstrate the role of the gut microbiota in driving metabolic changes, there's many ways you can do it. You can use germ-free mice and do some fecal microbiota transplantation. We do that very, very often in the lab. But of course, the problem with that is that you're using germ-free mice, and germ-free mice are not normal from the beginning. They have their limitations. And secondly, they're very, very costly and not very practical to use. But sometimes we still use them for small protocols. But in protocols where we use so many different types of animal models and different diets, we wanted to get some results quickly as well. We figured out one way to test the role of the microbiota was to see whether the change in the insulin resistance or obesity were actually preceded by change in the fecal microbiome. So why don't we do a very short-term protocol for about a few weeks, see whether that will change the fecal microbiome, perhaps even before the change in metabolism are occurring, the change of weight or insulin sensitivity. So we use again the same type of mice, male C57 mice. This time we only use the animals on the high-fat, high sucrose diet because we had no phenotype anyway, and the animals fed the low-fat, low sucrose diet, the healthy animals. So we used these animals on the high-fat diet, and we did a short-term protocol, only 2 weeks of a protocol. And as shown here, so the animals were again treated with the same treatment, except it was only high-fat, high sucrose for 2 weeks. And at the end, the difference is that we did fasting refeeding. I will explain to you why in a few minutes. Of course, we euthanized the animals to take tissues, and we did the blood sampling. And of course, we took fecal samples at the beginning. It's not shown here, but at the beginning and at the end. And the reason we did fasting and refeeding is that the time we were doing that, we were starting to collaborate with my good friend and previous student, Philip White, who is now at Duke Molecular Physiology Institute. And I was asking Phil, can we look at the metabolome, liver metabolome, and also maybe the plasma and the muscle metabolome of these animals to see whether there could be some hints about what is happening in terms of promoting metabolic disease. And Phil told us, well, we can do it, but it would be much better if we can do it in the refeeding state, because these metabolites will change much more into the refeeding state. So we did that. We did a short-term protocol. We look at the microbiome. We look at the profiling of short-chain fatty acid. And of course, we also did the plasma and tissue metabolomics in the refed situation. And we were very pleased to see that the gut microbiota was already affected after only 2 weeks. So this is the PCOA analysis showing the clustering of the microbiome in the different animals. You can clearly see that the animals fed the casein were clustered, the microbiome, the fecal microbiome clustered differently from the animals fed the mixed protein or the protein mix. When we look at the gut microbiota, of course, we didn't reproduce all of the changes we've seen after the chronic treatment with these diets, but it's interesting to see that already Alicrosia, as well as Tizarella and Intestimosas are already affected in a very similar way as what we have found in the long-term protocol. Akkermansia muciniphila is not on the list, but it was barely there. I think the p-value was 0.06. So we did some PCR to confirm that, and indeed, Akkermansia muciniphila was also downregulated or relatively less abundant to the microbiome of these animals after only 2 weeks of treatment. And I believe, again, here, this is the Shannon index, which is a marker of diversity. And you can see that diversity already increases in these animals with the protein mix after only 2 weeks. So most of the big change we saw in the microbiome, we could see in only 2 weeks of feeding the protein mix. What about the function in terms of producing short-chain fatty acid? So I'm showing you the data that were generated in the long-term protocol again, just to remind you what we had. And as you can see, this is the short-term protocol. This was 12 weeks. This is only 2 weeks. We see very, very similar change. Of course, after 2 weeks, we only look at the high-fat, high-sucrose-fed animals. But you can see we still have significant and very significant change, especially in isobutyric and isovaleric acid, as well as valeric acid, although less significant. And so we can reproduce these changes or this overproduction of branched-chain fatty acids in these animals after only 2 weeks of the protein mix-fed regimen. So what happens to the animals in terms of metabolic phenotyping? Well, these changes that I've just told you about the microbiome and the short-chain fatty acids, they were observed despite the fact the animals did not gain yet more weight. It's only 2 weeks of treatment. Body weight gain was the same. Glucose homeostasis was the same, as you can see here from the glycemia, the insulinemia, and as well the C-peptide levels. We did a bunch, again, of insulin signaling determination thanks to Vanessa Hood, but in that case, we couldn't see any difference in hepatic insulin signaling, which means that the change in the gut microbiota and the change in the production of the branched-chain fatty acid happened even before we could see metabolic defects. So in other words, they are not explained by change in body weight or insulin or hepatic homeostasis. So of course, what we wanted to look at with the collaboration of Phil White was, is it possible that within only a 2-week treatment period where we see these change in the gut microbiome, is it possible already that there's some change in the liver, in the plasma, or in the muscle in terms of metabolites that are crucial and are good biomarkers of metabolic homeostasis? And indeed, when we look at the liver amino acids, and some of these changes are also found in the plasma, by the way, but in the interest of time, I'm only going to show you the liver. We saw some increases in glutamine and glutamate, ornithine, asparagine and aspartate in the liver of the animals fed the protein mix diet. According to Phil, this is probably related to the fact that in the when you look at the amino acid composition of the protein mix diet, we actually have more glycine and more proline in the protein mix. That could explain probably the increase as well in the glycine, of course, in the liver of these animals. We also look at liver organic acid just to get a glimpse of the maybe the overcharge of the TCA cycle in these animals. And some of the change are almost significant or are significant. Clearly, we have an increase in many of these organic acid, which might be related again to change in aniline and aspartic acid as well. And most importantly, these changes were also very consequent or in agreement with the fact that acylcarnitines were not only increased in the liver of these animals, but even in the plasma. And these, of course, acylcarnitines are very good biomarkers of early mitochondrial overload and incomplete oxidation of fatty acid in the liver of these animals. So what we believe is happening is within already 2 weeks of feeding these diets, there's a change in the gut microbiota, in the branched chain fatty acids, and somehow this is linked to change in a metabolic homeostasis in the liver. We saw partial change as well in the muscle, but not as robust as that we've seen in the liver, suggesting that there is actually a reduced fatty acid oxidation in these animals. And that, of course, fits pretty well with the fact that brown fat activity seems to be reduced in these animals. So there's probably less fatty acid oxidation in brown fat, but probably also, of course, in the liver and muscle. Now, this is the slide that is probably the most important to try to put this thing together, because the model we had at that time was that maybe the branched chain fatty acids that are produced more in the animals of the protein mix diet, are these branched chain fatty acids inducing perturbations of glucose homeostasis? These are coming from the gut microbiota. They're probably reaching the circulation, but can they directly interfere with metabolic homeostasis? So to begin to address that directly, we use FAO hepatocytes and L6 myotubes in vitro, and we decided to do incubation of these cells with isobutyric acid and isovaleric acid. So in the hepatocytes, we're talking about overnight incubation with these two branched chain fatty acids. And what you see here is the production of glucose. So it's gluconeogenesis because it's coming from lactate and pyruvate. And as you can see, when you start to increase the concentration of these branched chain fatty acid, you start to have more production of glucose and with both type of branched chain fatty acids. And importantly, even when you put insulin in the system, of course, insulin will block hepatic glucose production and the EFL cells, but you can see that even in the presence of insulin, the branched chain fatty acids are still increasing the production of glucose. So what we believe is happening is that the overproduction of branched chain fatty acids in these animals for the protein mix is driving glucose production by the liver, is promoting insulin resistance in the liver. When we did similar studies in L6 myocytes or myotubes, we were not able to see any effects of the same concentration of the two branched chain fatty acids in muscle, which is pretty consistent with the fact that we could not detect impairment of insulin signaling in muscle in the animals fed the protein mix diet. Now, can we link this effect to the mTOR pathway? And so Vanessa, again, used her magic hands to do signaling work in the lab, and she looked at the ability or the potential effect of isobutyric acid and isovaleric acid on the mTOR S6 kinase pathway, using, again, phosphorylation of S6 as a readout of overactivation of this pathway. And what she's seen is that when we did a time-dependent study, we see that after only 1 hour of giving to FAO hepatocytes these different branched chain fatty acids, we see an overactivation, an activation of the pathway that is at least for 2 to 5 hours, clearly reaching 5 hours in terms for isovaleric acid, as you can see here. So there's an overactivation of the mTOR pathway that is not there after 24 hours. So this is a transient phenomenon, but we know that effects on the mTOR pathway can be seen, and the impact on insulin signaling can be driven by for many, many more hours. We've seen that before many years ago. So what we believe is happening is that These branched-chain fatty acids are produced by the gut microbiota in PM, in protein mix fed animals. They activate the mTOR pathway in the liver, and this is driving insulin resistance and increasing glucose production by the liver. Of course, the next step will be to do these studies in vivo with naive animals and to try to see whether we can reproduce these effects in an in vivo context. But in vitro, it really clearly shows that there's a direct impact of these intriguing molecules on insulin signaling and glucose production. So this is the model we are working on. This work is actually submitted for publication in a high-impact journals. We hope to know very soon if it's going to be accepted. But this is the model. So clearly, if you feed a high-fat high-sucrose diet, whether it is casein or a humanized protein mix, you will get, of course, insulin resistance, hepatic steatosis, and obesity. But what we found here is that if you really give to these animals the real proteins that a human eats, so not only casein, but all these mix of protein like beef, chicken, etc., you see a much greater production of microbial BCFA. This is connected to hepatic insulin resistance, more mTOR pathway activation into the liver. So we have this fatty acid oxidation alteration and increased gluconeogenesis. And if you wait for 12 weeks, you will actually get an expansion of the white adipose tissue and a reduction of brown fat, which is, of course, accompanied by an increased obesity in this model. But we believe that the production of BCFA by the perturbed microbiota is the early events that are producing perturbations of metabolic homeostasis in animals fed the protein mix. So again, this is the work of Beatrice and Noémie, but many people in my lab were involved into this work, and I would like to thank them. And also collaboration with Frederick Backhead and Marcus Talman from Sweden, and Philip White, of course, from Duke University for all the metabolomics, and I'd like to thank him again for that. So for the second part of my talk, I would like that we are looking beyond the gut, because until now I've been talking about the gut microbiome. But we recently published a paper that you might have seen in Nature Metabolism that is showing that microbes can cause metabolic disruption not only in the gut. So as just to introduce this new tale, I just want to remind you that, of course, this is taken from Benoit Chassain's paper in Nature in 2015, where he was showing us that as compared to animals treated with placebo, where in the intestine, you can see that the bacteria, which is highlighted here in red, labeled in red, are pretty far from the epithelial cells in the intestine because they're separated by the mucus layer, this green label mucus layer, which is protecting us from invading microorganisms. Well, if you treat animals with CMC or P80, which is well-known emulsifiers that is used by many companies, many agro-food companies, what you see is that these mucus protection is going down, is reduced after only a few weeks, and these bacteria then are coming closer and closer to the epithelial cells. You have a breach of the barrier, and not only bacterial fragments can reach, therefore, the epithelial cells and cross the membrane, but maybe also bacteria can do it. And there's been evidence already in animal models that you can actually label bacteria in an obesity model and find these bacteria present in adipose tissue, for example. But it still don't know if these bacteria are living in the adipose tissue in these animal models. But anyway, as you know already, for example, Akkermansia muciniphila can be neutralized, can be killed, and you can see still metabolic benefits with Akkermansia. It's probably the same with some pathogenic bacteria. They don't need to be alive, maybe to drive inflammation in metabolic tissues. So the idea here is that microbes can reach the circulation, probably translocate to metabolic tissue to trigger immune metabolic dysregulation. But what about human obesity and type 2 diabetes? There's very little known about the presence of these bacteria, especially in the adipose tissue in the liver. So we team up with many friends and colleagues here at Laval University, including Andre Chernoff, but also with Jonathan Schretzer at McMaster University, and folks from Vaomer, a company in Toulouse, which are leaders into looking at the amount or the type or the abundance of bacterial DNA in tissues that are well known to have very little bacterial DNA. It's quite challenging to isolate bacterial DNA and to analyze this from tissues containing very little amount of bacteria. And this work was published last month in Nature Metabolism. And I should really thank the two guys here, Fernando, who just became a father recently, so Fernando Anne, who started this project in my lab in Quebec and moved to Jonathan Schretzer's lab in Hamilton a few years ago and finished the work. And this also brilliant investigator in my laboratory, a postdoc, Benjamin Jensen, who joined my lab a few years ago. So those two guys really drove this project with the help of Thibaut Varin, a bioinformatician and a microbiome expert in my lab. Though there were a lot of people involved in this work, so I'd like to thank them already. So as the cover shows or suggests, we look at microbial DNA signatures in metabolic tissues, and we look at that in the context of type 2 diabetes. So with the help of Andre Chernoff, what we did is that we had cohorts of individuals coming to our center at the Institute, the Heart and Lung Disease Institute at Laval University. This is the hospital where my lab is. And Andre and I looked at different cohorts of people, and we're able to identify people that were either non-diabetic or diabetic. These are all people that came to the center because they had bariatric surgery, which of course is an opportunity for us to get access to tissues. So all of these individuals obviously are obese, as shown here by the BMI, the waist circumference, the waist to hip ratio, but only half of them were actually diabetic, and the other half was non-diabetic, because we wanted to know, irrespectively from obesity, if the presence of diabetes or the absence of diabetes was going to drive different changes in the bacteria that might translocate to metabolic tissues and even to the circulation of these individuals. So the only thing that differs between these individuals, there were men and women, is of course fasting glucose and HbA1c, so the diabetic state of those individuals. So the challenge with this kind of work, of course, is to make sure that what you identified is really bacteria in those tissues or the plasma and not contaminants, because of course we're dealing with, if you're dealing with the gut contains so much bacteria, this is less of an issue. But if you deal with fat, liver, and plasma, of course, you have to worry about the contaminants coming from the environment. So we add those tissues from morbidly obese, type 2 diabetic, or non-diabetic individuals. We had access to liver biopsies, omental, mesenteric, or subcutaneous fat from these different individuals. And of course, we had also access to plasma. And so we extracted DNA, not only from those tissues, but also from a bunch of different but very important controls. So from the tissue collection, we had different controls for the operation field throughout the tissue biopsies, the tissue manipulation and processing, of course. So we had controls for the lab, the biobank, we had swear and air controls, and of course the DNA extraction amplification step that were done at our partner in Toulouse, Vaomer. They also had their own controls. So all of the results I'm going to show you have been corrected for all of these negative controls to make sure that what I'm showing you today is really bacteria that were there over background contamination. And just to show you a little bit what it looks like, so this is a kind of a heat map of all the different, the catalog of all the bacteria we have measured in liver, mesenteric fat, omental fat, saturated fat, subcutaneous fat, sorry, and of course the plasma. And you can see already that there are different type of bacteria in the different tissues or in the plasma. This was expected. It was just to show you that we can detect bacteria in those tissues and that there is a variation in the expression or the abundance of these different bacteria in the different tissues in the plasma. But most importantly, what I want to show you here is that when you actually look at the gene copy number of all those tissues, it is clearly above what we can measure in all the negative controls that we looked at. So what I'm going to show you has been corrected for those negative controls, but it's just to show you that indeed we can detect a bacteria way over the background levels. The only worry, of course, is with plasma. Plasma was close to these negative controls. So we had to use very sophisticated bioinformatic tools to make sure that what we have seen in the plasma was really significantly increased over the negative controls. What I'm showing you here is the different indices of alpha diversity. So the observable to use, and especially the Shannon index. And as you can see, there were no differences except maybe between plasma and the liver. But in general, the different indices that we looked at in terms of diversity were not very different between the different tissues. Next, of course, we look at the clustering of these different microbiota clusters in the different tissues. And I think you can appreciate that mesenteric fat, for example, as well as plasma, were clustering differently as compared to the liver and to other adipose tissues in general. And when we look at the taxonomic profile of these different bacteria in the different tissues, of course, we also found some differences between the tissues. That was expected, of course. And what I wanted to show you as well is that not only there were differences in the taxonomy or the main phyla of these bacteria, but I'm going to show you 6 examples of bacteria that were clearly different between the different tissues. For example, Atrobacter was more abundant in the liver as compared to the adipose tissue and the plasma. And it was also the case for this particular Ruminococcus. This is a well-known gut commensal bacteria, but this is an environmental bacteria. And although it is an environmental bacteria, you see that there's still clearly more this bacteria in the liver as compared to the negative controls. And again, all of the data I'm going to show you today have been corrected for these different controls. When we have looked at, then we have looked at other type of bacteria. Here you see this is in the mesenteric fat. For example, we see more Bacteroidetes in the mesenteric fat, whereas in the omental and subcutaneous fat, we see more Enterobacter. So you see again, these gut commensals can be differently abundant in different type of adipose tissue. And of course, we also saw some environmental bacteria, such as this one here, very difficult to pronounce, Cluvera. This is an environmental bacteria that appears more into omental fat and saturated fat as compared to other tissues or plasma. And we have also Poleramonas, which is expressed or abundant or more present in the plasma as compared to tissues and the negative control. And this is again an environmental bacteria. So these tissues not only have gut commensals that probably translocated from the gut, but maybe even some environmental bacteria that we are consuming probably in our diet, and that maybe are not checked pretty well by the immune system in the gut and are crossing the barrier because these persons are obese. They have probably a reduced gut integrity. They have a leakiness of the gut, and these environmental bacteria seems to be able to travel to the adipose tissue and liver. Of course, we don't know at this point whether these are complete bacteria or just bacterial fragments, because what we look is 16S. So we see reeds, so they can be only fragments of bacteria. Even if there's a whole bacteria, they might not be living. So we still don't know if it's live, dead, or fragments of bacteria. That's the focus of future work. Now I'm going to show you what happens in terms of if you are diabetic or not. And first of all, just to show you again that there is no difference in the gene copy number that we saw between non-diabetic and type 2 diabetic individual. Again, all these individuals are obese. The only difference between them that we could see was their diabetic state. So you see no difference between non-diabetic and diabetic individuals for the gene copy number. When we use different indices of alpha diversity, the only reduction of alpha diversity that we have seen was in mesenteric fat. In the type 2 diabetic individual, there seems to be a less diversity in the mesenteric fat as compared to the non-diabetic individuals, but this was not the case for the other tissues. We look at beta diversity, of course, and if you look at the clustering of these different microbiota, we don't see really any significant change between or difference between the non-diabetic and the type 2 diabetic individuals. However, when we start to look at the tissue bacterial profiles in more detail, if you look at these different genera, for example, we do see differences in many bacteria between non-diabetic and type 2 diabetic individuals. And this is not very easy for you to see or for me to explain with these cladograms. So I'm going to use the opportunity that Patrice Cagny and Matthew Van Hoel have published in the same issue that we are publishing this work, a news and views about our paper where they look at, they have produced a very nice figure actually showing the effect or what kind of bacteria are different between the obese non-diabetic and type 2 diabetic individuals. And so what they have represented here is our finding is that there are some bacteria that can cross the intestinal membrane because of course these are obese individuals. And in the liver, we see some changes in this particular bacteria. For example, in the obese type 2 diabetic individual, we have more of this environmental bacteria called Aquabacterium. Also in type 2 diabetic individuals, we have a reduced diversity in the mesenteric fat, but we see also increased abundance, relative abundance of some bacteria, such as Enterobacteriaceae, and we have also a reduction of many bacteria, such as Fecalibacterium and Ronbutia, which are very well known for having impact on glucose and lipid metabolism, at least in animal models. And in the saturated fat, the only difference we saw in the diabetic individuals was this increase in sphingomyelin. And another very interesting one actually is Escherichia shigella. Shigella has been shown in humans to be increased in the blood of people with NASH, so associated with liver disease. And so we have also seen an increase in the type 2 diabetic individuals, obese type 2 diabetic individuals, as compared to non-diabetic individuals in the plasma. So overall, if I can summarize the data from this paper, is that we believe that not only gut commensals, but also bacteria coming from the environment, from the food or the water we drink, in a leaky gut, because in obesity, especially in a morbidly obese individual that we looked at, they have a leaky gut, they have impairment in gut immunity. And so what we believe is that they are translocation and encroachment of either live, dead, or fragments of bacteria that are reaching the liver, different type of adipose tissue, and of course the circulation. We haven't looked at whole blood. We should probably expect even to see more bacteria in the whole blood as compared to plasma. And what we think is that this differential bacterial compartmentalization in obesity is different, of course, between non-diabetic and type 2 diabetic individuals. And this will modulate the risk of developing type 2 diabetes and associated cardiometabolic disorders. Now, this was a rather descriptive study. What, of course, we need to do now, and we are starting to do, is to demonstrate really the mechanisms of action. So can we culture these different bacteria coming in the tissues? Can we isolate these bacteria from the tissues? Can we culture them, identify the species that are involved? Can we, for some of them, can we actually grow them, inject them into animals as probiotics, and see whether they are modulating glucose homeostasis? Because remember, some of the bacteria that were increased in obesity were actually more increased in non-diabetic versus diabetic individuals. That means that some of these bacteria might not be driving metabolic disease, maybe even protecting these obese individuals from developing metabolic disease. We don't know that. So we really need to look at individual bacteria, put them in animal models, and see whether we can modulate glucose homeostasis. So this is the next step that will follow up the study. But this is a lot of work, but it's going to be quite exciting to do this in the years to come. Again, this was published in Nature Metabolism last month. You can take a look. at it. And if you have any question, of course, feel free to write to me on Twitter, or of course at my email address, which is, I don't think I have it here, but it's very easy to find, or you can ask Phil White to provide my address to you. So most important slide for me is the acknowledgement. All this work was possible because of the incredible work of many students and postdocs, in particular Beatrice Choi, Noemi Daniel, Fernando Forato Ane, who is actually now in Jonathan Schetzer's lab in McMaster University, Benjamin Jensen, but also Thibaut Varin, Vanessa Houd, Bruno Marcotte for the studies in the cell lines, Philippe Saint-Pierre for helping us with the project with the dietary protein. This is our ninjas. They are the animal technicians in my lab that are so important to this work. Without them, we couldn't do anything. I have a lot of colleagues at Laval University that were very important in doing this work. Andre Chernoff, Andre Tremblay, Cécile Var, who is now back in Lyon in France, Denis Richard, Simon Marceau, Laurent Berthaud. Those two guys are bariatric surgeons. They really participated a lot in the study. I was so pleased of their excitement for the work. Thanks again, Simon and Laurent. From McMaster University, I've already mentioned John, and also Michael Surratt has been quite a good guy to make sure we're looking at the microbiota from the best angle. So thanks a lot, Michael. And we had friends also in Sweden and Toulouse and in Europe, so Frédéric and Marcus, who have done some measurements for us that I did not have time to show you today. And finally, our friends at Biomer really help us to measure those bacteria in tissues from the bariatric surgery patients. So Florence, Sébastien, Benjamin, thanks a lot for your work. And all the work I've shown you today was funded by the Canadian Institute for Health Research. I'd like to thank them for that. And I think that was my last slide. So we'll stop here. And of course, I'll be very happy to answer questions on Twitter or by email after I have some well-deserved lunch, because I'm starving now. And even if it's Easter Friday, I'm still gonna have something to eat right now. But I'll be looking forward to answer all of your questions. And I would like to thank again Phil White for organizing this. This has been quite an important, interesting series to watch. And I'm looking forward to see the next one. Thanks a lot, Phil, and everybody.